Hello viewers, today I am here before you to explain about the test cross. What is a test cross and what is its utility? As you know that for each character there are two alleles. For each character there are two alleles. Say for example, character is height and its two alleles are tall and dwarf. Suppose another character, flower color, there are two alleles, red and white. So for each character, since there are two alleles, so it is called allelic pair. One allelic pair, tall dwarf, another allelic pair, red and white. In this pair, one allele is dominant and one allele is recessive. For example, in the pair of tall and dwarf, allele for tallness is dominant and allele for dwarfness is recessive. In this pair, allele for red flower is dominant and allele for white flower is recessive. A dominant allele is represented by capital letter. Say for tallness it is capital T. For recessive allele, a small letter is given but of same alphabet. So for dwarf it is small t, not small d, but it is small t. For red flower it is capital R, for white flower it is small r. Right? So because each organism is diploid, chromosomes are present in pair. So each organism is having a pair of allele. If someone is having the pair capital T, naturally it will be tall. Another organism is having suppose capital T, small t, it will also be tall because capital T is dominant. But if someone is small t, so it will be dwarf because both are recessive. It means a dominant character, tallness, tall. It may be a pure tall, means homozygous tall, or it may be a hybrid tall or heterozygous tall. So a dominant individual may be either pure homozygous or it may be hybrid, means heterozygous. So if you want to test this dominant individual, the individual having dominant phenotype, physically dominant character you can say, what is its genotype actually? It is a pure dominant or it is a hybrid dominant. Then we have to perform the test cross. So this test cross is done to find the genotype of a dominant individual. Whether that dominant individual, the individual having dominant phenotype, that individual is pure or hybrid. To find the genotype, that individual is crossed with its recessive individual. So such a cross is called test cross. So here the definition of test cross comes when an individual of dominant phenotype is crossed with recessive individual. Why? To find its genotype. Then such a cross is called test cross. For example, we are having a tall plant. Tall plant is a phenotype. It may be a pure tall, capital T, or it may be a hybrid tall, capital T, small t. So if you want to test it, whether it is capital T or capital T, small t, so you have to perform its cross with its recessive, means dwarf. So there are two possibilities. If that tall plant is capital T, make its cross with dwarf, and in next generation 100% tall plants are produced. But if that tall, whose genotype is to be tested, if that tall is a hybrid, capital T, small t, make its cross with recessive, dwarf, then in next generation there are 50% tall plant and 50% dwarf plant. So if that tall plant whose genotype you want to test, you make its cross with recessive dwarf, and if the result you get 100% tall, so you should say okay, that tall is capital T, capital T. But if this result is not observed in next generation, 50% tall and 50% dwarf plants are produced, then your answer should be okay, that tall plant is capital T and small t. So in this way we can test the genotype of individual whose phenotype is dominant. Okay, so this type of cross is called test cross. Thank you very much.